just trying to manage a deadly syndrome, and now it's evolved into, well, one of the most sophisticated biological challenges we face.
It truly has. And the goal has shifted so dramatically.
Yeah.
We're not just talking about lifelong management through antiretroviral therapy anymore. That in itself was a massive, massive success, of course.
A revolution.
Absolutely. But the conversation now in the leading labs is focused squarely on achieving sustained remission or, the ultimate goal, complete eradication.
And that is the high-stakes vision we are diving into today. So we've pulled together material that covers global research collaborations, some of the most radical scientific breakthroughs.
Things like new vaccine platforms, gene editing.
Exactly. And crucially, the really complex ethical frameworks that have to guide all this work.
Our sources, they really map out this multi-pronged approach that's underway. The science is moving incredibly fast.
So by the end of this deep dive, you should have a really clear picture, a kind of roadmap that researchers are using to finally try and write the conclusion to the HIV epidemic.
Let's hope so.
Okay, so let's start with the most visible part of this fight, vaccine development. For decades, it really felt like we were just hitting a biological wall.
Oh, for sure.
We saw these early trials that delivered, promising but also just frustratingly modest results.
That frustration was very real, but I think it's important to see the bigger picture of those early efforts.
Okay.
So take the RB144 trial. It's often called the TIE trial.
Right, I remember that one.
It only achieved about, 31% efficacy, so not nearly enough for a rollout, but it was so far from a failure.
How so? If it wasn't effective enough, what did we actually learn from it?
It gave us crucial immune correlates of protection.
Okay, break that down.
It means researchers could finally dissect which specific types of antibody responses were linked to protection, even if it was only partial protection.
So it told us what to aim for.
For the very first time, yes, we understood what the immune system actually needed to do to block the virus. That one trial really defined the playbook for pretty much every vaccine candidate that came after it.
And I guess that groundwork is why this current generation of candidates feels like such a huge technological leap.
Yeah.
The speed and the flexibility are just...
Oh, they're transformative. What's fascinating is how the technology is built to directly address HIV's biggest challenge.
It's diversity.
It's massive genetic diversity. HIV mutates so rapidly that trying to vaccinate against just one strain, it's futile.
Which is why we're now seeing this focus shift to what they call mosaic immunogen strategies.
Exactly.
That's a serious name. So if HIV is constantly changing its disguise, how do these mosaic strategies work?
Well, they try to elicit a kind of global immune response. So instead of using a single viral protein sequence, they use these engineered sequences that are derived from multiple global HIV strains.
You're training the immune system.
You're training both arms of it, the antibody and the T cell arms, to recognize parts of the virus that don't change, the conserved regions. The goal is to build a response broad enough to neutralize pretty much any variant you might encounter.
So it's not just about getting a high number of antibodies.
No.
It's about making sure they're effective against a huge range of targets. And this isn't just happening with mRNA, right?
Not at all. And that's smart. You can't put all your eggs in one basket. Our sources point to a really diverse range of novel vector approaches.
Okay.
So you have platforms using adenovirus, which is pretty well established, but also newer, more sophisticated ones using things like cytomegalovirus, CMV, and different nanoparticle delivery systems.
So these vectors are like high-tech delivery trucks.
That's a great way to put it. Each one is optimized to present that HIV immunogen to the immune system in the most effective way it can.
And we should remember, even the big trials that get discontinued because of insufficient results.
Like some of the recent adenovirus ones.
Right. They aren't failures, not if you view them as part of this learning cycle.
No, because they immediately funnel massive amounts of negative data right back into the R&D pipeline.
And that helps refine the next wave of mosaic and mRNA candidates.
Exactly. Every single trial, successful or not, it tightens that target zone for the next one.
But okay, moving from